Anyway, good morning, everyone, and Happy New Year. I'm Tony Latai, president of the SFPM, and welcome to the first installment of the 2025 seminar series. I will introduce the speakers shortly, but first, just a little bit of business. With regard to these speakers, I would like to say that we like to schedule in advance. There are still openings in our 2025 schedule. We have an open date in November and an open date in December of 2025. And indeed, we are happy to start scheduling into 2026, horrifying as it is to even talk about a year that advanced. So if you are interested, please contact me directly or contact Aaron Jesser at Aaron at SFPM.io. Is that a working email address? Do you like that? Okay, Aaron at sfpm.io, or you can contact me. And we can keep this very healthy seminar series continuing. I would also, with two issues with regard to membership, we do value the membership experience, and we want this is a completely member-run society. We don't have a big administrative staff, so we do want we want this society to work for the members and also be done by the members. One point is that Diana Azam is looking for people on her membership committee, and the whole point of this membership committee is to make membership work well and to make it as easy and convenient as possible for people to join and make sure we're working for you. So if you are interested, please contact Diane Azam, or you can contact again through Aaron and he can put you in touch. That's Aaron at sfpm.io. And finally, with regard to membership, if you have not already done so and you are on this call, please renew your membership for 2025. It's very simple. You go to sfpm.io. In fact, I'll even share my screen. Oh, I can't. Never mind. It's not worth it. But just go to sfpm.io, and at the up top, it says Become a Member. And you can click on there, log into our membership portal, and renew very, very easily. I would ask that you renew your membership even if you are an associate member who is a trainee who is not paying any fees. But for the rest of you, please join. We run extremely lean. None of the board members earn any salary or any compensation for this, but we do need money to defray some fairly routine expenses, and the SFPM can't run without the membership fees. So with that, it is now my pleasure to introduce today's speakers. We are going to focus on regulatory issues, and I know that a lot of us like me, spend a lot of our daytime trying to work through technical issues and make whatever assay they're working on work better. And indeed, that's important. But I would have to say, I think for many of us, we've enjoyed a lot of progress over the years, and we have solved a lot of the technical problems that we want to. There are always more. I would say pretty categorically that what I've observed is the biggest impediment to the adoption of what we call functional precision medicine is no longer technical. It is purely and mainly regulatory. We're going to hear about that today. We've run into, and I think this may need some real deep delving at even the federal level, which perhaps the society needs to engage in. But I think these are new assays that regulators all over struggle to, with their enormous workloads, to encompass something that's brand new, that is different from measuring a sodium level of 140. So we'll see. We have with us today two people from St. Jude's. Kristine Cruz, who is Director of Scientific Research in the Department of Pharmacy and Pharmaceutical Sciences, and also a program director of the Residency in Clinical Pharmacogenomics. And she also directs the St. Jude's Pharmacotyping Resource. She will speak first, and then she'll be followed by Alejandro Molinelli, PhD, who is director of the Clinical Pharmacokinetics Laboratory in the Department of Pharmacy and Pharmaceutical Sciences at St. Jude's. If you have questions as you go through, we're a relatively informal group. You can put them in the chat if it's something urgent, and try to get the attention of me or the speaker, and we can call on you. Alternatively, you can wait till the end and put it in the chat or raise your hand, and I can call on you at the end. So with that, I will mute myself and I will welcome Kristine to start us off.
Thank you so much for that introduction, Dr. Latai. I think it was really well stated, so it sort of summed up kind of what's going on in this very new space, in this very translational research area where we work. So Happy New Year, everyone. We'll be talking again about regulatory issues, and these are some, very sort of logistical issues, some key points to understand in how precision medicine tests for patients are regulated. And so let me make sure I can move forward. So the agenda for today's seminar is shown here. We'll start by talking about who regulates precision medicine tests. We'll go through some definitions and descriptions, which is necessary for the conversation. The first half of the talk, I'll talk about the FDA regulation of assays, which the FDA considers devices. And then I'm going to hand over to my colleague, Dr. Molinelli. He'll talk about CLIA and CAP regulations of the labs themselves. And then we'll leave plenty of time to talk about the FDA final rule for laboratory developed tests, and also some time at the end for some conversation. So we know that precision medicine through laboratory diagnostics is really driving positive change in healthcare. And it's been estimated that more than 12,000 laboratories in the US run laboratory developed tests. It's also been estimated that approximately 70% of clinical decisions made on a daily basis rely on in vitro diagnostic testing. And so when we think about precision medicine, we know that genomics testing only gets us so far, because especially in oncology, where I practice, a minority of patients really have a definitive actionable finding with genomic testing alone. And so we recognize there's a need to extend these benefits to more cancer patients. And this is where functional precision medicine comes in. So functional precision medicine, by its very nature, is translational research in nature. It bridges the bench to the bedside, but it also brings knowledge from the patients back to the bench. And this is a continual process. So I've worked for many years under the tutelage of Dr. Bill Evans, who's now emeritus member at St. Jude, but really one of the fathers of pharmacogenomics and of translational research. And Dr. Evans has always said that translational research relies just as heavily on learning from the patient and bringing that knowledge back to the bench as it does from pushing out research to the patients. So as you're working through this process with your own test, it'll come a time when you say, when is the right time to seek approval for your test? Or really, logistically, when is the right time to bring your test to patients and offer it for clinical care? And what's the road forward for that? And so that's what we're going to be talking about today. And so for a case in point, just want to share with you briefly the functional precision medicine test that we're conducting in our lab. So in our lab in the Department of Pharmacy and Pharmaceutical Sciences, we have developed a novel test, and we focus this test on children with acute lymphoblastic leukemia. We can obtain leukemia cells from patients at the time of diagnosis or at relapse, and we test a panel of 20 drugs. We expose the patient's primary cells to a panel of drugs, and using fluorescence imaging, we measure the sensitivity or resistance of the patient's cells. And so our group, along with our collaborators, has shown that the results of this test really corresponds to survival outcomes and to patient outcomes. And so we're ready now to take this, until now it's been a research test, and we're ready to take this to clinical care and ready to take it into prospective clinical trials. And so we're really navigating that process right now. And so this brings us to the topics of today. So who regulates the safety and efficacy of functional precision medicine testing? So there's kind of three different categories that we can think about. So first of all, the tests themselves are considered devices by the US FDA. And so it's the FDA or the EMA in Europe that regulates tests. And the test does require approval before it's used for patients, and the FDA is interested in the analytical and clinical validity of your test. And then as far as the regulation of labs, in the US it's CLIA or CAP, or the EU states that regulate most labs. And CLIA is going to ensure that the lab meets minimal technical and personnel standards. And then lastly, there's reimbursement. So CMS or the EU states regulate reimbursement. And here's where the clinical utility of the test is going to be evaluated. And so it's a lot to navigate. And for the purposes of this talk, we're not really going to focus on the reimbursement because that's really outside the scope of today's presentation. We'll have enough to talk about just focusing on the first two categories. And so the first half of the talk, I'll focus on FDA and some definitions and descriptions of the process. And then Dr. Molinelli will talk about the regulation of labs, secondly. So first, let's talk about some definitions. So when you're talking about an assay, it's important to distinguish between clinical validity and clinical utility. And I see both of these terms used in the publications that are being published, but really to break down sort of the spectrum of measurements that can be reported. I'm going to break them down for you in categories. So first of all, going from left to right, we need to, of course, focus on analytical validity. So, and this is something that you probably have dealt with quite a bit in your lab. So this is referring to the ability of your test to accurately and reliably measure the analytes of interest. And this is typically documented in a validation report done by your lab, and it's reported through accuracy, precision, analytical specificity, and analytical sensitivity. And that's pretty clear and pretty non-controversial, and we're all pretty familiar with that. But then moving forward, when we think about clinical validity of a test, so this is evidence that the test reliably predicts a condition in a defined target population. So for example, for our test, it's in a defined population of children with ALL. The clinical validity must be shown before any results can be used for treatment decisions. And the measures here include really for a quantitative assay for establishing a cut point, and then measuring the clinical sensitivity and clinical specificity, as well as, if it's appropriate, positive predictive value and negative predictive value of your test. And this is where valid scientific evidence from well-controlled studies is needed to establish clinical validity. Then moving forward, the next step is clinical utility, which really is kind of even moving forward to provide evidence that the test meaningfully improves patient health outcomes when it's used to inform and support clinical decisions. And here's where functional precision medicine tests could be categorized for use for treatment response or for diagnosis. Precision medicine tests could be considered companion diagnostics to go along with a pharmaceutical. And this is where it gets pretty tricky, as you can imagine, because the test results of the types of assays that our labs are offering most likely indirectly affect patient outcomes, because what they're influencing really are the clinical decisions of the clinicians, which in turn affect patient outcomes. So teasing out how to show the evidence for that can get pretty ingrained in the details. So as far as the FDA is concerned, the FDA is not looking at clinical utility. The FDA is focusing on analytical validity of your test and clinical validity of how it's being used. Excuse me, how it's being used. And so we're going to focus on those two. So just to start with a couple of definitions here that will help our conversation. So the Food and Drug Administration of the US is the agency within the Department of Health and Human Services. It's tasked with protecting public health by ensuring that drugs, vaccines, biologic products, and medical devices are safe and effective. And here is where an assay, like the one that's offered in our lab, most likely like the one offered from your lab, is considered a medical device by the US FDA. So the FDA is codified by Title 21 of the Code of Federal Regulations. And you might be familiar with the IND regulations for new drugs and biologics. And these regulations are in 21 CFR Part 312. The regulations for devices are separate. So, and they're spelled out completely in 21 CFR Part 812. And so this is really the regulations that we're going to be pointing to for medical devices. So the FDA has a particular definition of a medical device, and it can be found in Section 201H of the Federal Food, Drug, and Cosmetic Act. It's a pretty broad definition, and it says that any instrument, machine, contrivance, implant, in vitro reagent that's intended to treat, cure, prevent, mitigate, diagnose disease in man. And so here is where the FDA considers our assays to be a device, a medical device. And so where they're evaluated, just to kind of map this out for you, so that everyone sort of understands where this is. So the FDA has 3 major medical products divisions. If you're a clinical trialist, you might be most familiar with CDER, the Center for Drug Evaluation and Research. There's also the Center for Biologics Evaluation and Research, or CBER. But where devices reside are in the Center for Device and Radiological Health, or CDRH. And this is the division that's really going to be evaluating laboratory assays as devices. So the FDA has a particular definition about an investigational device, and very simply, an investigational device is a device that's the object of an investigation. So if you're taking it through a clinical trial, it's an investigational device in the eyes of the FDA. And so we often hear the term IDE. And so just to break down what an investigational device exemption, or IDE, is, IDE refers to the regulations under 21 CFR Part 812. And an approved IDE means that the IRB, the local IRB, and the FDA for significant risk devices has approved the sponsor's study application, and that all requirements under this Part 812 are met. So very simply, an IDE is a regulatory submission that permits clinical investigation of a device. So if you're going to be doing a clinical trial of a device, an IDE is typically required. And one important concept is something that we've talked about with our local regulatory affairs office, and that's the concept of significant risk of a device. So medical devices and IDEs have to consider the level of significant risk during the development and clinical trials. And a significant risk device is defined as one that presents a potential for serious risk to the health, safety, or welfare of a subject. And actually, the determination of significant risk is done at the level of the IRB, and it's actually the determination of significant risk versus non-significant risk is made by the sponsor, proposed to the local IRB, and the IRB has to approve or disapprove whether the level of risk is significant or non-significant risk for a device trial. And so that means that as you're taking medical device through clinical trials, you have to consider risk management and mitigation, as well as concepts of design control. And these are all laid out fairly clearly.
Can I interrupt for a moment? Just
Yes.
because we entered exactly that circumstance.
Yes.
And as a matter of fact, it wasn't just our IRB. We actually were confronted by the FDA because our clinical trial required an NDA as well as an IDE. Mm-hmm. And it was determined by the FDA, not by our IRB, that it was a trial of significant risk. Which I completely disagree with their adjudication, but in that case, the adjudication was made by the FDA, not just our home IRB.
Yeah, thank you for sharing that, Tony. So we had conversations with St. Jude's IRB, and the thing that we were talking with our IRB about is, what is the risk to patients if your assay comes up with the wrong result, right? So if you're going to be making a diagnosis or choosing a therapy based on the results of that, what is the risk to the patient if the wrong choice is made, if the wrong decision is made? And so that's those are the things that the IRB has to grapple with when we think about significant risk. And so as far as taking your device through trials, in the FDA's eyes, there's basically 3 categories, and I plotted them out here. So if we think about all clinical trials that involve devices together, you can start to break them out. And going, really sorry about this, but going from right to left is kind of going from simpler to complex. So the FDA will consider some studies to be exempt from the IDE regulation. And studies that are exempt will be studies on devices that, for example, are already a commercial device, and you're doing a trial on something that's within the scope of its approval. Then those trials will be exempt from the IDE regulation. But studies that are subject to the IDE regulation, there are significant risk investigations and non-significant risk investigations. And so for non-significant risk studies, these studies do not require submission of an IDE to the FDA, but there are some abbreviated requirements such as labeling, IRB approval, informed consent, monitoring, and some reporting. But for non-significant risk studies, the IRB is serving as the FDA surrogate for reviewing and approval and for continuing review of those studies. If it's determined that your device trial is a significant risk investigation, then the full requirements are required, and the study really cannot begin until an IDE is submitted and approved by the FDA. And this is where probably several of us are kind of navigating through which category your device clinical trial will fall under, and then which requirements have to be followed. And so this is sort of a very high overview of how to walk through that. So I do have some good news, and that is that device study design differs from drug and biologic study design. So if you have been involved in, for instance, oncology clinical trials, you're probably very familiar with the phase 1, phase 2, phase 3 requirements for a pharmaceutical. Device studies are a little bit simpler. So what FDA requires for a device approval is one pilot study, which is can be a fairly simple feasibility study. It can be exploratory, it can be the first time in humans, and it can involve a very small sample size. And then only one pivotal trial beyond that is required. And that pivotal trial will determine the safety and efficacy. But in contrast to pharmaceutical research, which often you need thousands of patients for the pivotal trials, the one pivotal trial for a device approval can be much smaller and could be hundreds of patients rather than thousands of patients. And then plus or minus, there could be some post-marketing studies to be done as well. But what's required is one pilot study and one pivotal study for a device. And there is a division which is available to answer questions about device investigation. So the Division of Industry and Consumer Education, or DICE, is available to answer questions. And there is a website which is affectionately called Device Advice. And you yourself, as someone from a researcher who's looking to take your device through clinical trials, can submit a question through the website, which is on the Device Advice website. And you can ask a question about, should I seek an IDE? Should I not? Where should I start? And the Device Advice staff will get back to you within 48 hours with at least a preliminary answer to your questions. And so there is some help to sort of how to get in to start navigating this process through Device Advice. And just to walk through a case study of our lab's pharmacotyping assay. So we're looking to take it from a research assay to a clinical assay. And so we've started, of course, with the analytical validity, and we've completed that. It took us close to a year of doing the validation studies that were required. So we've documented in a validation report for pharmacotyping test of a 20 drug panel, which we test our live patient cells with. And so that part is complete. And so where we're transitioning now, we're moving into clinical trials for our assay. And so we've determined that we are going to start with a fairly simple pilot study with a fairly simple feasibility question as its primary objective that's shown here. And it's just to evaluate whether our test can be performed in the clinical setting for our population of interest, which is children with ALL, and the results can be returned to the patient's medical record. So after our fairly small and fairly simple pilot study is done, then we'll move to a larger pivotal study to really start to collect the clinical validity data and move forward in that way. So we don't anticipate the need to show clinical utility. Again, this is beyond the scope of what the FDA is looking for. So with one pilot study and one feasibility study, we should get the data that we need moving forward. And so that's a really quick synopsis of FDA regulations. And so what I'd like to do is hand over to my colleague, Dr. Alejandro Molinelli, who'll talk about the regulation of labs and also get into the FDA LDT final rule. And then we'll have time for conversation with each of you. So go ahead, Alejandro, I'll stop sharing. And Alejandro, you're still muted.
Yes. All right. You should be seeing my screen now. All righty. So thank you for that, Chris. So as most of you are familiar, in the United States, wait a minute. There we go. Okay. In the United States, in order for a laboratory, a clinical lab, to perform testing for diagnostic or treatment purposes, it needs a CLIA certificate. So the purpose of my talk is to talk a little bit about CLIA. So this is going to be fairly US-centric. I'm going to talk about the CLIA regulations. I'm going to talk a bit of its relationship with the College of American Pathologists. I'm going to have a couple slides on ISO 15189, the international regulation for the international standards for clinical labs, for medical labs, and I'll talk about the FDA final rule. So in essence, CLIA, and it's called CLIA 88, it's Clinical Lab Improvement Amendments. The original act, the CLIA Act, was from 1967, but it was not until 88 that these amendments really strengthen the regulatory requirements of CLIA. So CLIA provides minimum standards that the federal government enforces, and Centers for Medicare and Medicaid Services, CMS, has primary responsibility for administering the CLIA programs. Now, you've all heard of the College of American Pathologists, Joint Commission, all those inspections. So all those are deemed designees. These are approved accrediting organizations under CLIA. So you can have a CLIA certificate if you just go straight to CLIA, and that's or a certificate of accreditation if your accreditation actually comes from one of the approved accrediting organizations. And I have a list here of all of them. I highlighted in yellow the two more important ones, because those are the ones that are more relevant to probably academic hospitals, like ours, the College of American Pathologists and the Joint Commission. And that would be the Joint Commission Lab Accreditation Program, not their hospital accreditation program, which is separate. But you can see that there's more than the CAP as an kind of approved accrediting organization. So talking about CLIA itself, so CLIA is the federal law, but it actually, multiple agencies actually share authority to implement the CLIA standards. So the FDA categorizes tests based on complexity. CMS actually issues the certificates. They also administer, they also have the primary responsibility for managing the CLIA program. And actually the CDC develops technical standards and lab practice guidelines. The CDC also manages something called the CLIAC, the CLIA Advisory Committee, which is made of mostly of lab professionals, and it meets twice a year. And actually, they listen to the advice of the CLIA committee, advisory committee. I want to show a little bit of that relationship. So I mentioned that College of American Pathology, and our lab is accredited. We have a CLIA certificate of accreditation, and our accrediting agency is the College of American Pathologists. So I'm going to show here is just kind of how does that look on paper. So the 493.1443 is the CLIA standard, straight out of the regulations. And you can see that, well, for CAP, all these deemed agencies, they can have standards that are equal to or more strict than CLIA. You can say that CAP is definitely more strict than CLIA, here's just an example. This would be the lab director qualifications. How do they look in the CLIA standard, and how do they look on a CAP checklist item? So CLIA prescribes personnel qualification standards and technical standards for the lab. It actually has a very strong emphasis on the role, responsibilities, and performance of the lab director. This is now taken from CAP. CAP actually has a director assessment checklist. So CAP has a lot of discipline-specific checklists. They would be chemistry, microbiology, molecular pathology, so on and so forth. They have a general checklist, they have a common checklist, they have a lab director specific checklist. And actually the performance of the lab director has to be set and complied with. So the lab has to set what acceptable performance of the lab director is, and they have to assess it annually. Not just of the lab director, but also the testing personnel. And actually that's one of the things that can be a pitfall for labs, in that it's not just the technical personnel that need competency assessments, but also the lab director needs a competency assessment. And you can see that the lab director is responsible not only of proper performance, but if anything is going wrong with the lab, it's gonna get cited too on that director checklist. So the lab director needs to be involved in the lab. The lab director needs to know everything that's happening in that lab. He needs to ensure that all the personnel are qualified to work in that lab, and that you maintain all the standards necessary. The other part of CLIA would be the technical part, and here's just an excerpt. I kind of copied here several of the important technical sections of CLIA. These would be most of these are part of the quality system requirements from CLIA, everything from procedure manual, test equipment, maintenance and function check, calibration, test records. So it gets very detailed. I highlighted in yellow, establishment or verification of method performance specifications, because this is a big one. And this is, I'm kind of starting to dive a little bit into what's the FDA final rule, and maybe explore, we can speculate of the why the standard rule is, the final rule is coming along. So you have establishment verification of method performance specifications. So let's dip a little bit deeper into that. So that's actually something that Chris mentioned on one of her slides. So if you have an FDA cleared test, so this is not a lab developed test, this is something that you're purchasing from a manufacturer, something that they took through the FDA and they're marketing with FDA approval. When it gets to your lab, you don't just put it in the instrument and start running it. You still have to verify that the performance matches what the manufacturer says as to precision, accuracy, reportable range, reference interval verification. If you have a lab developed test, instead of verifying the performance, you have to establish the performance parameters. So you have to do the same experiments, but to establish the performance. You also have to add a few more things: analytical sensitivity, specificity, and this is the big one, any other performance characteristic required for test performance, for example, stability, any other thing. So we felt there's a lot of, it's a little bit looser as to what it is. Is it similar to what the FDA is requiring of a manufacturer? Not necessarily. Is it bad for patients? Not necessarily. That's actually a big area of debate. So what's the role of the FDA? So as I mentioned, the FDA CLIA, the FDA is one of the agencies that is involved in CLIA for the longest time. You know, I really like this slide. This image comes from NIH, and it shows the world of medical devices and how FDA regulates. And you have in vitro diagnostics, and you have that circle with lab developed test. So lab developed test the FDA has practiced what's called enforcement discretion. So they say that they have decided, kind of just to put it that way, not to enforce lab developed tests. Now, this has been done for several decades. There's a lot we can argue about it. The big change that happened was that last spring, the final rule for lab developed tests was passed by the agency, and now we have to comply with it. I'm going to have some details of what that final rule means at the end of this section. The other big role the FDA has played is test complexity categorization. So they'll categorize tests as wave, moderate complexity, or high complexity. At least there's some of the criteria they use to gauge test complexity. Most of the tests that we do in our laboratory, and actually I forgot to mention that our laboratory mostly does therapeutic drug monitoring and clinical toxicology testing. So we use techniques that we use include automated immunoassays, but also mass spectrometry. So we're a fairly mass spectrometry heavy lab. All the mass spectrometry tests are actually high complexity. Most immunoassays that don't require preparation steps would be considered moderate complexity. If you have preparation steps, they actually step up to high complexity. So most of what we do in our lab is high complexity. So very quickly, I want to touch on ISO 15189. So ISO 15189 is an international standard that specifies requirements for quality and competence in medical labs. It actually focuses on having a quality management system. That quality management system should align the lab's mission, goals, and objectives to those of the organization. It should focus on the patient, and it should encompass all the activities, everything from personnel, technical requirements, suppliers, should encompass every aspect of the lab operation. So one thing to mention is that in the United States, ISO 15189 is optional. However, you can seek ISO 15189 accreditation in the United States. There's several, I'm going to call them brokers for that. Actually, the College of American Pathologists is one of them. They actually offer 15189 accreditation. It's separate from their lab accreditation program. They're not the only one. The American Association of Lab Accreditation also can accredit under ISO 15189. But I do want to mention that it is optional in the United States. I did mention a QMS. QMS is very important under ISO 15189 accreditation. And I have here two definitions, one from the FDA and one from ISO itself of what a QMS is. The way I see it like the movie The Matrix from the late 90s with Keanu Reeves, where the Matrix permeates everything. So that's how you see a QMS. It should be permeating every aspect of what you do. Your personal qualifications, your procedure manuals, your quality assurance practices should kind of affect every process in the lab. And it should be aligned with your lab's mission and with what the product you're delivering. Here's a nice table that I adapted from a CLSI publication with the differences between quality control, which is basically method control specific to that test that you're doing, QA, which would be process management, focusing on pre-analytical processes or focusing on the analytical part or focusing on post-examination. And what the QMS is, which is that all-encompassing kind of system. All right, want to compare very quickly the difference between in the red box, what a CAP inspection is like, and what an ISO inspection, as we've experienced it with CAP, with the CAP 15189 process is. So under the CAP inspection process, it is based on CLIA regulations. It is lab specific. So I showed a couple of the CAP checklists. So it goes by discipline specific checklist. It focuses on procedures, stringent criteria set by CAP experts, accreditation every 2 years, and in the year in between, you do a self-inspection. Also, for the CAP inspection program, it is peer inspections. So we, as practicing medical lab scientists, have to get inspected every 2 years, but we also have to inspect a peer institution every 2 years. Some of us do it more often because there's often a need to do that. On the other hand, the ISO, and I'm going to focus on this ISO process as viewed through the lens of how CAP is delivering the ISO 15189 program. It is one set of standards. It focuses on process. The lab needs to show they have that integrated QMS across the organization. You actually get a professional auditor that's furnished by CAP. It's not peer inspections, and you actually keep the same auditor for the 3-year cycle. It is a 3-year accreditation cycle with an internal audit every year. And on year 1, you get a full inspection, year 2, a management inspection, year 3, a technical inspection. So you're basically getting inspected every year for the 15189 program, and you also get your CAP accreditation. So you actually don't do one or the other. You have to do the CAP program. If you do 189, you do both. All right. Before we delve into lab developed tests, one last thing is that because you see this a lot, and you say you see it in marketing materials that say that a test is CLIA approved, and that to me is wrong because CLIA does not approve tests. CLIA regulates labs that perform testing on specimens for diagnostic or treatment purposes. The lab needs to ensure your lab has analytical validity, as Chris mentioned. CAP also has clinical validity requirements. None of this is furnished to the FDA. This data, this validation data that you have in your lab is usually assessed by your inspection team every 2 years when they come for the on-site inspection. So there is no CLIA approved test. There's a CLIA certified lab, but not a CLIA approved test because CLIA is not approving tests. Only the FDA is approving tests. And one of the things that the FDA requires, what the CAP requires of labs, is that if we use a lab developed test, we have to put it on every patient report that we're using a lab developed test. That's not cleared by the FDA. However, the laboratory is CLIA accredited, and we can perform this test for diagnostic or treatment purposes. So that brings us to the final rule. So final rule was passed last spring. It has 5 stages. This is fairly foreign for us that work in medical labs. It is a whole new world. Stage 1 compliance starts in just 4 months. So on May of this year, we should have ready our medical device reporting requirements, corrections and removals, and complaint files. By next year, registration, listing, labeling, and compliance with investigational use rules that already exist. It's just that now we have to comply with them because many labs do not. Stage 3, quality system requirements that would include design controls. This is the hard one. This is actually already a heavy lift. Complying with full QSRs, design controls, purchasing controls, acceptance activities, CAPAs, and records. And then the big one for 27 would be pre-market review for high-risk LDTs, where you have to seek pre-market approval, or pre-market review for low and moderate risk LDTs, which would be the 510K or de novo process. So is this fairly burdensome? Huge. Is there a lot of uncertainty surrounding the final rule? Yes, there is. There's two legal challenges that are outstanding, one from the Association for Molecular Pathology, another one from the American Clinical Lab Association. That litigation is ongoing. I'm not going to speculate as to the outcome. There could also be legislation challenges to the final rule. There could be executive challenges to the final rule with an incoming administration. So there's a lot of uncertainty. Is the final rule incredibly burdensome for labs? Yes, it is. Incredibly, hugely burdensome. It's going to be very costly, and it will likely lead to less access to testing for patients. Is it going to meaningfully increase the quality of the test that we offer? I don't know. I would be speculating. Maybe it will, maybe it won't. There's a lot of reason as to why the final rule came to be. They're casting a wide net. There's always kind of good actors and bad actors out. The way the final rule is kind of written, it casts a wide net and it affects everybody. So we'll see what's going to happen with the final rule. I have one more slide. It comes from our colleagues at ARUP, one of our reference labs. And it's not a simple rule to understand. This matrix actually helps a lot with compliance with the different phases of the rule. It also explains a little bit some of the areas of continued enforcement discretion. There's a lot of detail and nuance to this rule. So we can have multiple webinars on it. So I'm not going to go into more detail on it. I do have some resources on the final rule, starting from the FDA LDT website, a CLSI white paper. CAP has a series of 6 webinars to go into the different aspects of the rule. MSACL, mass spectrometry applications for the clinical lab, that's an association I kind of work together with. We have our own kind of resources page for the mass spectrometry community. If you're interested in litigation updates, I actually put a link there. There's a lot of information about the final rule that we can go. We can take some of the questions, but there's a lot of uncertainty. There's really, all we know is that the final rule is still the law of the land, and we have to comply with phase 1 by May 6th. That's what we know. Other than that, we'd be speculating. All right, that's all that I have. Want to thank particularly the drivers of this of this project, Dr. Yang and Dr. Brown and the team in Pharmacy and Pharm Sciences, some other of our colleagues at St. Jude that have helped us, and I guess we'll go ahead and go to the question section.
Great. I want to thank both of you for really an outstanding talk. Just addressing a question I saw just came up on the webinar. This is recorded, and members, active members of the SFPM will have access to this entire recorded talk. It'll be up on the SFPM website under the seminar series. I want to make a couple editorial comments before we open this up. With regard to some of the politics of this, everything you just said about the FDA and the final rule is, I think, a bit of a moving target. Under the prior administration, HHS indicated defending the FDA final rule as one of its top legal priorities. It is not clear to me that the incoming administration will share the same legal priorities at HHS. That remains to be seen. I will say that you are absolutely right. This is burdensome and obstructive. It may surprise you to know that there are some organizations, some diagnostic organizations, that are strongly in favor of this. For some, it's because it provides clarity. The cynic in me tells me it's not merely for clarity that they are doing this, but also to clear the competitive landscape, because big providers, I'll let you decide which these might be, but big diagnostic providers see a signal advantage in regulatory burden in that they can handle it, they can absorb it, you cannot. So you are fighting people. It's not as if all diagnostic players have the same interest in this. Obviously, this is way too complicated. This is an example, of sort of the airplane being built while they're flying it. There's the idea that we have CMS and the FDA and multiple threads of regulation even within the FDA is absurd. I'm going to ask one question and then maybe go through some of the questions in that. My question is, we've heard really about as good as you can do in an hour about these different regulatory regimens. When do you need an IDE and when do you need to do it via CLIA? My impression is that if you're doing a clinical trial, an IDE suffices whether or not the test is being performed in a CLIA environment, and you actually do not need additional LDT final rule certification if you have an IDE. Is that correct?
I think that's correct, as you said it. Actually, as Alejandro walked through the process for the LTT final rule, just checking a box that you're complying with IDE regulations is part of the requirement. So it's kind of all kind of rolling together, right? And I don't know if Alejandro, if you have another way to express that.
No, I think that would be accurate.
Okay. There's a question if this needs to be randomized by Rita Fior. Totally makes sense. You absolutely do not need any randomization. No one at any of this level of regulation is concerned about whether this thing works or whether it works better than other things in the clinic. Surprisingly, they don't care. They care about all the other analytic things and the other validations that Alejandro and Kristine were telling were telling us about. Now, are there any other? I don't want to ignore active questions of people who are actually online right now. I don't see any hands up. Are there any? If I'm missing you, please speak out.
Hi, can you hear me?
Just barely.
Oh, okay. Hi, I'm Kelly Gordon. I just wanted to add a comment just because it might be helpful for you. I've worked a lot in companion diagnostics and have just started helping some companies developing functional precision medicine assays. And so there's lots of lessons learned from companions. One of the things about the study risk determination, just from my own experience, I often leverage the pre-IND and the streamlined IND pathway for study risk determinations, because what I've found in some situations, CDER and CBER would be more likely to give you a non-significant risk than if you take it to CDRH or the IRB. And it can really help to get that excerpt from that study risk determination up front to take to your IRB so they don't put you on clinical hold. So just a thought that might be helpful to people.
So just to clarify, Kelly, you're talking about risk. This is something, again, that confuses me. I don't know if we're going to merge with any clarity here. Kristine was talking about the IRB performing the risk determination. I'd gotten muddled, and I was pretty sure the FDA had gotten involved in ours, whether we wanted it to or not. And now you're saying that, in fact, you have some choice in the matter, and you can ask for risk determination either from your IRB or from the FDA.
Absolutely. So both can do it. And in my experience with CDXs, I actually prefer to go to FDA. I've had IRBs be more conservative. It really depends on your relationship with like a local IRB versus a central IRB. But my preference is actually to go to FDA. I find that I am more likely to get non-significant risk. Now, all contexts are different, but just some feedback from my perspective.
And now, do you know, so we went down that route and the FDA gave us what I think is a ludicrous significant risk determination. Is it possible to go back to our IRB now? Or I think I saw something in the comments suggesting that that is not possible.
So it's not possible. So FDA trumps IRB, but what is possible is I have helped. So I'm a consultant and I've helped a lot of clients that maybe didn't answer the study risk determination questions quite clearly or provide enough rationale in the original submission. And I have helped them go back. So they may have, for example, just provided a little bit too light detail in terms of their study risk determination, and they got an IDE because FDA often will go to the most conservative approach for a new assay, especially. But you can revisit it. Again, it depends on context. They really are looking sort of, as Kristine said earlier, that the consequences of a false negative and false positive and what that really means in regards to the study design.
Kelly, you say you're a consultant in this field, and that might be that your services would be useful to our members. If you contact me, I'd be happy to post your information on our SFPM website so that interested parties could contact you.
Absolutely. Thank you.
Okay.
Yeah, I just had one point for clarification on your CLIA versus non-CLIA requirement. Maybe people on the call can address, though, the typical approach is that if any information is going back to a patient for determination of care or something like that, that it would be performed in a CLIA lab. So I think maybe people can clarify what they were talking about before, where they're performing assays and doing development.
Yes, well, I can answer. If a test is being used for what you call diagnostic or treatment purposes, and that test result is going to go to the medical record, and the practicing clinician is going to use that result to make a decision, the test has to be performed on a CLIA certified lab.
Or IDE if it's being performed under the aegis of a clinical trial.
Yes.
Yeah,
I just have a
don't know that the
clarification.
information can go back to the patient though, right? It could be used by a provider or something, but I don't know. I'm not so
I will
sure.
say,
I've
what I
never
think,
heard any exceptions to
so
the
Keith,
CLIA.
I've talked to a lot of different people who've encountered this, and I think you are treading on a gray area where it actually depends on individuals on whatever panel you're dealing with. So you can imagine situations where information is returned that mandates a treatment decision. That's one thing. There's other situations where information is returned and the clinician and the patient can choose to roll it up in a ball and throw it in the garbage. They don't need to act on it whatsoever, but it's available to them. That I would put to you as a gray area right now, exactly how regulation of that formed.
I just had a quick question for Kelly, clarification. My understanding, based on people who've done this process, is that in order perhaps to prevent IRB shopping, if an IRB determines something is a significant risk, you really can't go back. You can't go to another IRB and ask them to determine it's not a significant risk. You get one bite at that apple at least.
Yeah, you can't go to another IRB, but you can go to FDA. So absolutely. Yeah. So you just take the IRB correspondence and go to FDA. Usually in that situation, I'll go to CDRH because I want to have the device people basically reject IRB's opinion. But I always proactively go first, if possible, to CBER and CDER again, just because I do find that because they're looking at the study context as a whole, not just on the assay. They're looking at the patient population design, et cetera.
You would think.
Thank you.
There's a question. Many reagents from Colin Paul. Many reagents that are used for functional testing are specifically labeled research use only, RUO, sometimes accompanied by a statement that they are not for use in diagnostic procedures. How are these regulated when they get used in regulated IVDs? Kristine, Alejandro, when do you want to handle that?
I can mention something to that effect, and that's actually part of so if you use research use only reagents, so now you have an LDT. So some of our LDTs actually use research use only reagents. So up to now, we've been using that through the current regulations revolving lab developed tests. How are we going to do that going forward because at some point, once you go to phase 3, we're going to encounter the purchasing controls. So that's when it's going to become really sticky because we have to then validate those reagents ourselves. The vendor we're purchasing them for is not doing their due diligence. They're just selling them research use only. So now we'll have to validate. That's one of the big questions we have regarding RUOs, regarding analyte-specific reagents that are labeled RUO once you encounter that phase 3 with the purchasing controls. So that's actually one of the, well, there's many, but that's one of the many areas where labs and actually lab associations have sent extensive lists of questions to the FDA. So far that I know of, we haven't gotten response from the FDA. But I can say that at least the ADLM, Association for Diagnostics and Lab Medicine, they have sent an extensive number of questions to the FDA that haven't been addressed. That's actually one of them. But there's several dozen questions there.
Right. Thanks. I think we're going to need to bring this to a close. I think this is an active area. I'll point out that we have a working subcommittee led by Keith Ligon and Bruce Yeager that is purely involved in regulation, and I encourage you to contact either of them or me or Aaron to get hooked up with the committee if you're interested in this further. I think this is a very complex area. It all feeds through the FDA. And I would say a lot of the decision-making is done by individual human beings at the FDA. And to put it mildly, their priorities do not align with our priorities all the time. And that is one of the things that I think we struggle just on a very individual basis, that regulators often have different priorities than the people who are trying to get things regulated. So I want to thank Kristine and Alejandro again for really just a great talk. It'll be available shortly as a resource on our website, on our members only section. Thank you very much. And I look forward to seeing you all in February. So long.
Great discussion. Thank you all.